Medicine Without Cruelty presents The Code of Life DNA. Now, I fell in love with biology and the life sciences because of DNA, first discovered by Rosalind Franklin back in 1953. Through her work, she began to unravel the mystery of how life is transmitted from cell to cell, from generation to generation. And that discovery of DNA structure sparked a revolution in the biological sciences and technology and expanded knowledge in many other fields. Based on the structure of DNA, the new science of molecular biology was born, leading to prevention, diagnosis, and treatments in ways that were unimaginable in 1952. The advances in identification and analysis of the genetic code based on Dr. Franklin's work have produced breakthroughs and has continued to help us understand human life. After studying and training and working within the field of medical genetics to diagnose individuals with genetic diseases, I can say one thing for certain: no animal can model human diseases, nor can they be used to judge whether a medicine or chemical will harm us humans. It can't be used, and the way forward is non animal methods. Just to remind ourselves why we're here, the greatness of a nation and its moral progress can be judged by the way its animals are treated. So, welcome to the Code of Life DNA. Today, we're diving into what makes each of us unique at a biological level, and that's our DNA. DNA, or deoxyribonucleic acid, and don't worry, we do not need to know that big long word, we just need to know DNA. But that's called the blueprint of life because it contains all the instructions that allow living beings to grow, function, and survive. Imagine it as a complex code that holds the recipe for who we are, from our physical traits to our inner biological processes. In a previous video, I talked about cells and how they are the smallest units of life, and how the nucleus acts as a control center housing our DNA. But today, we're going deeper, exploring not just how DNA guides life, but how small differences in this code sets us apart from animals. These genetic differences are key to understanding why animal testing falls short in predicting human outcomes and why human focused personalized research is essential for the future. So let's start by looking at what DNA is and why it's called the code of life. Now, DNA first discovered in 1953, as I said, by Rosalind Franklin, and that black and white photo is one of the first photos of DNA. And as you can see, it's made up of two long strands that are twisted into a shape called a double helix. You can call it a twisted ladder, where the sides of the ladder is made up of sugar and phosphate chemicals, and the rungs or the steps of the ladder is made up of four chemicals, also called bases. These four bases are called adenine, thymine, cytosine, and guanine, but we just call them A, T, C, and G. They always pair up in a specific way. So A always pairs with T, and C always pairs with G. DNA is a set of instructions, and each gene within our DNA acts like a specific recipe, instructing the body on how to make proteins, which perform crucial jobs like building tissues or sending signals. That unique order of A, T, C, and G in our DNA determines those traits, from eye color to how our body responds to diseases. This sequence varies across individuals and species, which is why humans are different from animals, despite sharing much of the same DNA. Now, biological evolution refers to the change of living things over time. Charles Darwin introduced this to the world in 1859, and he proposed that all living things descend from their ancestors, but that they can change over time. And it occurs as a result of natural selection, which is a theory that in any given generation, some individuals are more likely to survive and reproduce than others. Now, when it comes to animal testing, Darwin's theory of evolution has long been held as a justification that all animals, including humans, share a common ancestor, and that shared genetic similarity is reason enough to use these animals to test as models of humans. But as we'll see, that the small differences between animals and humans have fundamental implications for us, and animals are just simply not suitable for testing. I want you to think of DNA as a cookbook that holds the instructions or recipes for building and maintaining a living being. Each gene is like a specific recipe detailing the steps for making proteins that contribute to traits like eye color, height, or metabolic processes. Many animals share a large percentage of their DNA with humans, meaning that the general instructions could be similar. For instance, humans and chimpanzees share about 98 to 99%, like I said. Imagine following a recipe that's mostly the same for two versions of a dish. One might call for, I don't know, almond flour, and the other might call for oat flour. While the recipes are largely identical, this small swap in ingredients can lead to different textures or flavors in the final dish. In genetic terms, even a small change like swapping one DNA base for another, called a mutation, can have significant effects on how a protein is built or functions. These tiny genetic differences explain why humans and animals, even with similar genetic recipes, develop differently. For instance, a mere 1% difference between human and chimpanzee DNA affects brain structure, immune function, and susceptibility to diseases. And I should mention here that chimpanzees have been banned from animal testing for a number of years. I believe the last chimpanzees to be moved out of a laboratory into a sanctuary happened in 2006. So that was a long time ago. However, monkeys that are still used in research, particularly macaques, and they share about 90 to 93% similarity to us humans. But that 7 to 10 difference, what I'm trying to say is that difference causes fundamental distinctions between our biology and their biology, meaning that they are incapable of modeling human diseases or protecting us from potential harmful chemicals. A slight ingredient swap in our DNA can alter how our cells behave, making animals unreliable for predicting human responses. And that's why using human specific models is critical for accurate research, as they capture these small but impactful genetic nuances that animal models just can't do. So, if you're not convinced, let me talk about how animal DNA differences matter in research. Firstly, even the slightest genetic variation can cause animals to metabolize drugs differently from humans. For example, a drug that might be safe for rats or mice might be toxic to humans because of the differences in enzymes that break down these substances. For example, aspirin is toxic to cats but completely safe for humans. Penicillin is dangerous for guinea pigs but has revolutionized medicine for humans. These differences arise because of each species having unique genetic factors influencing how their cells react to different substances. Also, it's important to know that animals often have immune systems that function differently from human immune systems due to differences in genetic background. And that can lead to inaccurate predictions for human reactions to infections or treatments. For example, mice are frequently used in immunology research, but their immune responses to diseases such as cancer do not mimic those of humans. Not to mention the high failure rates due to these genetic differences. Now, drugs that are seemingly safe and effective in animals fail in human clinical trials 92 to up to 95% of the time. This failure is often caused by these small genetic differences, leading to massive biological discrepancies in how animals and humans react to the same drugs. Now, this is really important because we see time and time again how the devastating effects of animal testing has affected humans. For example, there's been studies where 6 different animal studies in 4 different species showed that this arthritic drug Vioxx, for example, had protective effects against heart disease, but it caused 320,000 cases of heart attack, stroke, and cases of heart failure across the world. 140,000 of those cases became fatal and they died before this drug was pulled in 2004. And so this can be disastrous. The high failure rate is not just a statistical issue, it leads to wasted resources, increased costs, and delays in discovering safe treatments for humans. And so we need to look towards human relevant models which provide results by mimicking human biology accurately. And finally, personalized medicine is an approach which tailors medical treatments to individuals based on their unique genetics, as well as perhaps their lifestyle and environment. It is impossible to replicate a human genetic environment in animals. Now, there are fundamental differences both at the genetic and cellular level. What does this mean? Even if humans and animals share many of the similar genes or recipes as we call them, how those recipes are expressed, meaning turned on or off, varies significantly between species. And now gene expression controls how cells grow, respond to the environment, and perform functions. If you think about it like a switch in a control panel, while humans and animals may have similar switches, in animals certain genes may be turned on or off differently, leading to distinct biological responses. For example, humans and mice might both have immune response genes, but the way in which these genes are activated changes, making mice respond to diseases and drugs in ways that humans simply do not. Now, believe it or not, cells do talk to each other. Human cells communicate through signaling pathways using specific proteins or chemical signals to talk to each other. Animal cells have similar pathways, but the signals involved or proteins can be slightly different or work in different ways, and that impact can be massive because differences in cellular communication mean that animals and humans can react very differently to the same drug. For example, a drug that targets a human pathway might miss its target in animals or might activate unintended responses, making the drug appear safe in animals but risky for humans. Now, proteins and enzymes matter greatly because they're like molecular machines, you could call them, in cells that help carry out various tasks from breaking down food in your body to repairing your DNA. Human and animal cells often have different forms of the protein, meaning that they react differently to drugs and environmental factors. And a nice example of this is the enzyme which breaks down caffeine, for example, is less active in dogs compared to humans, meaning caffeine is quite toxic to dogs because they can't break it down as easily. This difference illustrates why testing on animals does not predict how humans will respond to substances. And these fundamental differences mean that animal testing misses side effects or benefits that would appear in humans. By focusing on human relevant models that use actual human cells and tissues, researchers can get an accurate picture of how drugs will behave in the human biology, improving safety and effectiveness for real world applications. Just a case study now, targeted therapy is a type of cancer treatment that focuses specifically on the unique features of cancer cells. So, unlike traditional chemotherapy, which affects all rapidly dividing cells, targeted therapy is designed to identify and attack cancer cells with minimal damage to all of your normal cells. And it works by the fact that every type of cancer has very specific markers or targets on or within the cancer cells. These targets are often proteins or genes that cancer cells really need to grow, divide, and survive. And so targeted therapy drugs are developed to find and interact with these specific targets, blocking the cancer's growth pathways and effectively killing the cancer cells. It's really important when complex diseases like cancer, where people respond very differently to the same treatment, and animals just simply lack that diversity found in humans. And so, a promising application of organoids in cancer research is the use of patients' fresh tumor tissue to establish patient derived cancer organoids for personalized medicine. Those applications can be wide ranging, from preclinical modeling, so modeling the disease before it even enters further clinical trials, to drug screening, discovery, personalized medicine, discovery of more cancer biomarkers so we can develop more targeted therapies, among many other applications. So, just to sum up here, animal testing fails in 92% of cases to predict human outcomes, wasting resources and lives. Human specific models are not just ethical, they're accurate, reliable, and vital for real medical progress. The future of science is humane, innovative, and effective. Animal testing belongs in the past. It's an archaic science and it needs to be gotten rid of. Thank you very much.